So today we are going to learn about enzymology. So enzymology is the study of enzymes. So in this video we will be covering the basics of enzymes and the classification on the basis of what the reaction they catalyze. So we will be looking into these topics. So without any further ado, let's get started. So first of all, we need to know about what actually enzyme is. So we keep on hearing that enzymes are biocatalysts. Right? What this mean is that they enhances the rate of reaction. Right? So in biological terms, they say that enzymes increases the rate of reaction by decreasing the activation energy. Now, if you want to understand in a layman language what is the role of an enzyme is, then let me give an example of a student and a teacher's relationship. That a student, when he reads a book, he finds it difficult to understand the whole concept by himself, but when a teacher comes and teaches him, he finds it quite easy, right? So here the teacher actually is the enzyme, right? I hope you all have understood. So we have seen that enzymes increases the rate of reaction by decreasing the activation energy, right? So this was the very basics thing. Now when enzyme undergo the whole reaction, they do not change themselves. Do not undergo any change, right? And because of this property, they can be reused again and again, right? So one more thing to note down here is that they are specific in nature. That is, the meaning of this is that they only bind to where they have to undergo the reaction. For example, DNA polymerase, they only undergo in the replication process, right? And there are various, we will be looking into the classification, we will be easily to understand why they are specific in nature, right? So this was the very basic thing. Now, enzymes, when they are, like if they want to undergo any catalytic activity, they need non-protein components as well undergo various, and not all the enzymes but a few enzymes, right? So those non-protein component are called cofactors. Now before understanding this, all enzymes, we need to know that all enzymes are proteins, but not all proteins are enzymes. Okay, remember that. Now there is an exception here where RNA acts as a ribozyme. Okay, there is only one exception so far given. Okay, all the enzymes are protein in nature. Okay, so now coming back to here, that in inactive state, when enzymes do not undergo any catalytic activity without the presence of this non-protein, they are called as apoenzymes. So they are the inactive enzymes in general, but when they get a cofactor, they become holoenzymes. Now one more thing to note down here is that these cofactors can be divided into two categories. One are known as, I'll write it here, factors. One is known as metal ions, right? For example, if we use Mg2 plus, etc. And another where an organic molecule is used, and they are known as coenzymes, right? So this was the very basics of what actually enzyme is. So now let us move into the classification. On what basis are enzymes classified into? Okay, so now let us understand the classification of enzyme. Now sometimes enzymes are classified into two types. One is extracellular and another is, guess what, intracellular. Now you might be wondering what are these, and from the name itself you can understand those enzymes present in, not in, out. Okay, extracellular, outside the cell, right? Present outside the cell are called extracellular enzymes. So all the digestive enzymes come under this. Okay, similarly intracellular enzymes, those enzymes which are present within the cells are called intracellular enzymes. Now not this is the actual classification. Enzymes are classified also on the basis of what reaction they catalyze. So we'll be looking into that. So now we need to understand that enzymes are classified into 6 types. What are they? O, T, H, Lil. Don't worry. This is actually a way to understand, memorize, right? All this reaction. So first one is oxidoreductase. Second one, okay, O stands for oxidoreductase. Second one, transferase. Third one, hydrolase. Fourth one, guess what? Lyases. Fifth one, isomerase. Sixth one, guess what? Ligase. Now apart from this, there is also a seventh one which is known as translocase, but we are not going to include it in this video. Okay, so now we'll be understanding a little bit brief about all those reactions and where enzymes on base of the reaction which they catalyze. So first of all, first comes is the oxidoreductase, where both oxidation in these reactions, both oxidation and reduction take place, right? As you can see this example, where I have taken an example of a step in glycolysis, right? So here A refers to a pyruvate, B refers to NADH, and as you can see here are the electrons. So what actually happens, as we know that oxidation and reduction both process can, and it's reversible, it can be in either process, right? So what happens, it either transfers, it either accepts the electron or releases the electron, right? So here the enzyme which is responsible is lactate dehydrogenase, right? So as we can see that the B has the electrons and which is being transferred to the A, right? And it can happen even in the opposite direction as they are reversible reactions. Now coming to the second category, which is the transferase, and here what actually happens, the transfer of the functional group occurs, right? And here I have taken example of translation process, where the amino acid, as you can see this reaction, A represents amino acids, B represents tRNA, and X represents an amino acid residue, right? Let us take a chain, right? So this is an amino acid chain. The tRNA transfers this functional group to the amino acid chain, and the reaction, which is catalyzed by the enzyme known as peptidyl transferase. Now from the name itself we can understand transferase, it actually transfers the functional group, right? So what will happen, these amino acid residue will join with this chain and it will form a big polypeptide chain, and further the tRNA will be free of the amino acid residue, and we have seen this in the translation process. So now comes hydrolysis. So from the name itself again we can understand that hydrolysis is occurring here, the breakdown of the molecules, right? So here mainly water will be involved, okay, and the enzyme which will be involved. Now let us see this example first, okay, after that we will understand. So this is an amino acid, this is a water molecule, and the breakdown of this amino acid happens. So first we have a lysine and amino acid alanine chain, okay, and with the help of water and an enzyme it breaks down into simple separate compounds, lysine and alanine. So now the enzyme which is responsible for the breakdown in this reaction is the serine hydrolase. So this are the three OTH, okay, and now let us move further. So now moving further, let us understand the rest three, which so we have seen oxidoreductase, transferase and hydrolysis. Now let us see about lyases, isomerases and ligases. So in lyases what happens, again breakdown, dissociation happens here, okay, but here there is no involvement of water molecules, okay. So let us take an example. So what reaction, so we have a reaction A where it gives two products, A plus B, right? So breakdown of this product happens. So this is an example of urea cycle, where arginosuccinate breaks down into an arginine and a succinate with the help of an enzyme called argininosuccinate lyase. It's simple as that, right? So now moving further, isomerases. Again from the name itself you can understand isomerase, isomers, okay. So in isomerase what actually happens is there is a change in the structural change, okay, not in the chemical formula, okay. So here we have the example of glucose 6-phosphate which converts into fructose 6-phosphate with the help, okay. So the enzyme here involved is the phosphoglucose isomerase, okay. So far we have covered OTHLI. Now last but not least it is the ligase. So I'll be writing over here what actually ligase is, okay, because we have to explain. So we have this reaction A plus B, okay, will give a product. So let me explain. So here we have DNA, okay. We'll see about the role of an enzyme called DNA polymerase, okay. No, not DNA polymerase, DNA ligase, okay, DNA ligase. So what it actually does, here we have the reaction, right? A is this strand and B is this strand. Now in order to stick those ends, stick this strands, we need an enzyme, right? So here the enzyme DNA ligase acts as a you can say a glue, right, in simple words. So what will do, it will join this unoccupied space. Okay, so that's the function of ligase, where it acts as a glue. So apart from this, we forgot to mention about what actually EC numbers are. EC numbers refers to enzyme commission numbers. They are nothing but from a specific number given to the particular enzymes, right? So they're like this, for example 3.1.1.1. so what this portrait is, this is the class of the enzyme, this is the subclass, this denotes the subclass, and this number denotes to the particular enzyme. That's it. And one more thing to note here, which I forgot to mention earlier, about prosthetic groups, right? So what are prosthetic groups? As we have seen about cofactors involved, right? So the apo enzyme and the cofactors together make an holoenzyme, right? But the cofactors which are tightly covalently bound to each other are known as prosthetic groups, right? Whether it can be a metal ion or an organic molecule. If they are tightly bound together, okay, which they hardly disassociate, they are called as prosthetic group. You just need to remember that. And now after this, we'll be also looking into about what all factors are involved in the factors affecting the enzymatic reactions, right? What all factors are seen, and we'll see in another videos about that. So far in this video we have covered what actually enzymes are, right? On what basis they have been classified, right? On the reaction with the catalyze, and we have even seen on a different basis like extracellular and intracellular with examples. And that's it. Thank you.